Strip away everything. What is a living cell actually made of? You are at the atomic level, mostly empty space, but the parts that matter. 4 families of molecules: biological molecules, carbon-based organic compounds found in every organism on Earth, from bacteria to whales. Carbon is the backbone because it is the only element that forms 4 stable bonds, letting it build the chains, rings, and branches that complexity demands. Here is the elemental fingerprint. Every biomolecule carries carbon, hydrogen, and oxygen. But then, things diverge. Proteins and nucleic acids also need nitrogen. Only nucleic acids carry phosphorus. And some proteins contain sulfur, hidden inside cysteine and methionine residues. 4 families, 4 jobs. Carbohydrates store and deliver energy, build cell walls. Proteins catalyze, defend, transport, and give you your shape. Lipids seal every cell and stockpile long-term fuel. Nucleic acids carry the instructions, manual, and copy it every time a cell divides. How can 2 identical molecules in this biomolecule family make both bread and wood? Same formula, same atoms, completely opposite consequences. The culprit is 1 hydroxyl group on 1 carbon. Glucose in solution coils into 6-membered pyranose ring. Everything hinges on carbon 1, the anomeric carbon. When C1 hydroxyl points downward, you have alpha glucose. When it points upward, you have beta glucose. Chain thousands of alpha glucose together and you get starch, digestible food. Chain thousands of beta glucose and you get cellulose, wood. 1 atom, 2 worlds. Now shrink the ring. Ribose is a 5-membered furanose ring, the sugar backbone of RNA. Deoxyribose has one tiny difference. The OH at C2 is stripped down to just an H. That missing oxygen is why DNA outlasts RNA by thousands of years. Less reactive, more stable, better for long-term storage. Adding another key point about this biomolecule topic. Why can your liver release blood glucose in seconds, but a tree trunk stands for centuries? When 2 monosaccharides collide and expel a water molecule, you get a glycosidic bond. That is condensation. The bond type, alpha or beta, and exactly which carbons connect determines everything about what the polymer does. Amylose chains alpha glucose in neat alpha 1 and 4 spirals. A helix you can actually coil iodine inside, turning blue-black. Glycogen takes the same backbone and adds alpha 1 and 6 branch points every 8 to 12 units. All those branches mean more free chain ends for enzyme to attack at once. Your liver can flood glucose into your blood within seconds of stress. Cellulose takes the opposite bet. Beta 1 and 4 bonds flip each glucose unit 180 degrees, creating dead straight chains that lock together with hydrogen bonds into cables of extraordinary strength. No enzyme in your gut can crack those beta linkages. That is why you cannot digest wood and why plant cell walls hold up skyscrapers of cellulose. Our next build focuses on amino acids and proteins, 20 building blocks. How do you get a million different proteins from just 20 ingredients? Every amino acid shares the same skeleton. A central alpha carbon gripping 4 things. An amino group, a carboxyl group, a hydrogen, and an R group. That R group is a variable. Change it and you change the amino acid. 20 different R groups, 20 amino acids. String them together and something irreversible happens. The carboxyl group of one reacts with the amino group of the next, expelling a water molecule and snapping into a peptide bond, condensation. This bond is planar, it cannot freely rotate. That rigidity is what forces the chain to fold in predictable ways. A chain is a polypeptide, the sequence of amino acids. The primary structure is everything. Change one amino acid and you can change the shape. Change the shape and you change the function. The R groups drive all foldings, all polarity, all interactions. Tracking this protein polymer, our next build focuses on peptide folding. How does a flat chain of amino acids become a working 3D machine? A gene writes the instructions. Those instructions spell out a sequence of amino acids. That sequence, held together by peptide bonds, is the primary structure. It is the source code. Everything that follows is the code running. The backbone does not lie flat. Hydrogen bonds between the C double bond O of one residue and N single bond H of another force local regions into alpha helices, tight right-handed coils making 3.6 amino acids per turn, or into beta pleated sheets where separate strands zip together with H bonds. That local organization is secondary structure. Now the whole chain collapses into its final 3D shape, tertiary structure, held by hydrogen bonds, ionic bonds, hydrophobic interactions, van der Waals forces, and disulfide bridges between R groups. This shape determines the enzyme active site, the antibody binding pocket, everything. And if two or more folded chains associate, that is quaternary structure. Hemoglobin carries 4, 2 alpha and 2 beta subunits. Moving to this lipid family, our next build covers amphipathic cell boundaries. Why does your cell membrane assemble itself without any instructions? A triglyceride is simple. 1 glycerol backbone, 3 fatty acids, 3 ester bonds, and 3 water molecules expelled. Saturated fatty acids have no double bonds, straight chains that pack tightly and solidify at room temperature. Unsaturated fatty acids have C double bond C kinks. They cannot pack, so they stay liquid. That is the difference between butter and olive oil. Now remove one fatty acid and attach a phosphate group instead. You just created a phospholipid, a polar hydrophilic head and 2 hydrophobic tails. Put this molecule in water and it solves its own problem. The heads push towards water, the tails hide from it. The result, spontaneous, no energy needed, is a bilayer. Every cell membrane you have ever had assembled itself this way. Steroids do not look like other lipids. 4 fused carbon rings, completely rigid. Cholesterol sits in your membranes and controls how fluid they are. It is also the raw material for testosterone, estrogen, cortisol and bile salts. The same molecule that clog arteries in excess is essential for life in the right amounts. Testing this biomolecule polymer, our next build covers genetic code. How does a molecule store enough information to build an entire human being? The unit is a nucleotide, 1 pentose sugar, 1 phosphate group, 1 nitrogenous base. chain thousands together with phosphodiester bonds. 3' hydroxyl group of one sugar to 5' phosphate group of the next. And you have a strand of nucleic acid. The base sequence is the message. DNA is a double helix, two antiparallel strands coiled around each other, held by hydrogen bonds between complementary bases. Adenine thymine with 2 hydrogen bonds, guanine cytosine with 3. Guanine cytosine pairs are stronger, harder to separate, which is why DNA with high guanine cytosine content is more thermally stable. RNA is the working copy, usually single-stranded, uses ribose instead of deoxyribose, and uracil instead of thymine. 3 flavors. Messenger RNA carries sequence out of the nucleus. Transfer RNA physically brings amino acids to the ribosome, and ribosomal RNA forms the ribosomes itself. Analyzing this inorganic biomolecule, our next build covers cellular water. Why is a molecule this simple the single most important molecule for life? It looks trivial, 2 hydrogens, 1 oxygen, but the geometry matters. Water's bent shape 104.5 degree combined with oxygen's greed for electrons create a polar molecule. Partially negative charge on oxygen, partially positive charge on each hydrogen. That polarity means water molecules cling to each other and to each polar molecules through hydrogen bonds. Those hydrogen bonds give water remarkable properties. High specific heat capacity. It resists temperature change, buffering your body temperature. High latent heat of vaporization. Evaporating from your skin carries enormous heat away. Cohesion and adhesion. Water pulls itself 10 meters up a tree against gravity, and it is an unmatched solvent for every ion and polar molecule your biochemistry depends on. The ions matter too. Sodium and potassium generate your nerve impulses and muscle contractions by rushing across membranes. Calcium is the trigger signal for muscle contraction and neurotransmitter release. Iron nestles inside hemoglobin's heme ring and physically grip oxygen. Magnesium sits at the center of every chlorophyll molecule, making photosynthesis possible. Hope you liked the video and got your concept cleared. Subscribe our channel for more such tutorials.